Hello everyone, welcome to the class. So from today we'll be learning about the new concept, that is fine structure of gene or organization of genes and chromosomes. In this module we'll be learning about several topics like gene families, homologous genes, pseudogenes, genome, the parts or the portion of the gene, chromosome and its organization, histone modification and its resultant effects or consequences. So like this. So from today we'll be discussing about a different topic which will lead us to advanced concepts like DNA replication, RNA synthesis, recombination, protein synthesis, control of gene expressions in our future classes. So let's start with the basic concept, that is gene. Gene was the word which was first used by Wilhelm Johansen in 1909. So what are these genes? Genes are the functional segment of genetic material. As you know, our nucleus, the major backbone of the whole cell, or the major controlling center of the cell, or the center of attraction of a cell is nucleus, right? In nucleus we had discussed about there are chromatin networks, or there are thread like structures which consist of genetic materials, that is known as DNA, deoxyribonucleic acid. In viruses it is different, that is the genetic material is RNA. So where are these genes present in them? So in the genetic material, so these genes are the functional segment which has a particular function to perform, which has the expression or a character or a trait to be expressed in the organism. So it is locatable region of the genomic sequence. It is a unit of inheritance. So these points help us to know it is functional, it is locatable and it is inheritable. It is associated with regulatory regions, transcribed regions and or other functional sequence. So these genes are not a single entity, they have some regions, sub-regions in them. So genes are transcribed into mRNAs, that is messenger RNAs, which encodes the protein. Number and size of genes. Total gene number is known for several organisms. The bacterium with the smallest known genome is M. genitalium, has approximately 470 genes. Here M suggests Mycoplasma genitalium, which has approximately 470 genes. Number of genes in a eukaryote varies from 6,000 to 40,000 genes. The number of genes in bacterial genome is proportional to genome size. So the number of genes in eukaryotes does not correlate with the genome size or the complexity of the organism. As I mentioned that gene is not a single entity, or it does not have a single regions, so they have different portions in them. So the major portions of gene is non-coding DNA and coding DNA. So non-coding DNA interrupts the relatively short segments of coding DNA. So this non-coding DNA interrupts the relatively short segments of coding DNA. Coding DNA which has a particular function to perform. So it is a coding segment. The coding sequence are called exons and non-coding sequence are called introns. Here in this picture you can see the presence of introns, exons. So exons are the coding DNA. The coding DNA segment is interrupted by the non-coding DNA. So let us now learn about introns. So what are these introns? As said earlier, it is an intervening non-coding sequence. Any nucleotide sequence, it is a nucleotide sequence which is represented in primary transcript of gene but not present in the finally processed form. There is a process where these introns are removed and exons are preserved. Okay, so there is a process you will come to know in future classes. So in the introns are present in the primary transcript of genes. Before removing, there will be presence of intron. After that, these introns non-coding sequences are removed so that the coding would be easy and continuous. So these introns were discovered in 1977 by Philip Sharp and Richard Roberts. Many genes like interferons, histone genes, ribonuclease genes, heat shock proteins, that is HSP genes, G protein, coupled receptors lack introns. So all these lack introns. Okay, not required for gene function in eukaryotic cell. So introns are not required as it is non-coding. It doesn't code for anything and it does not required for gene function in eukaryotic cells. Introns are not found in most genes of lower simple eukaryotes, found rare in genes of prokaryotes. Some common introns are GUAG introns, AUAC introns, group 1 introns, group 2 introns, pre-tRNA introns and RKL introns. So these are some examples of introns. What are introns now? Introns are non-coding sequence which do not code for anything. They interrupt the coding segments of the DNA. Coming to gene families. So gene families are the set of genes. They are the set of genes with sequence homology descended from a common ancestral gene is known as gene family. So it is a set of genes with the sequence homology. Something common in the sequence is descended from the common ancestral gene family gene. Members of gene family may be closely related or even identical, may be clustered together or dispersed on different chromosomes. These gene families are highly variable in size, sequence, diversity and arrangement. Let us now know about the classification of gene families. So classification of gene family is based on diversity and functions of genes within the family. One is multi-gene families and another one is gene superfamilies. Multi-gene families are the group of genes with related overlapping functions and arise essentially from gene duplication. And the gene superfamilies are the group of related multi-gene families, group of common origin with non-overlapping functions. Here you can see non-overlapping functions. So there is no particular not overlapped function. So here we can see related overlapping functions. So there are subtypes like simple multi-gene families, complex multi-gene families. In simple multi-gene families are the families in which all the members have nearly identical sequence. So the gene families which are belonging to the simple multi-gene families will be having some identical or nearly identical sequences. Example, rRNA. Complex multi-gene families are the families in which members are similar to sequence but different for the gene products to have distinctive properties. Example, mammalian globin genes. Here you can see the organization of globin gene families in several mammalian species. So now we will discuss about the different types of genes. So genes is homologous gene, pseudogenes. In homologous genes we can see paralogous and orthologous. The gene is having two regions, intron regions and exon regions. Gene can be classified as pseudogene. In pseudogene it can be conventional gene and process genes. Homologous genes, genes present in same organisms is known as homologous genes. So under homologous there are two categories, that is paralogous and orthologous. So the genes which are present in the same organisms that arouse through duplication and divergence is known as paralogous genes. Homologous genes, the genes present in different organisms that arose through speciation and divergence is known as orthologous genes. So the genes, as already discussed, it has a intron region which is a non-coding DNA sequence, exon regions which is coding DNA sequence. Gene is also of type pseudogenes, where pseudogenes means false genes. Genes are inactivated because of accumulation of mutation is known to be conventional genes. Pseudogenes, which are the genes derived from reverse transcription of mRNA and integration of that reverse transcribed DNA is known as processed genes. So genes are of two types here discussed: homologous gene, pseudogenes. Homologous genes, the genes present in same organisms that arose through duplication and divergence is known as paralogous genes. The genes which are present in the different organisms that arose through speciation and divergence is known as orthologous gene. The pseudogenes, which are the false genes, the genes activated because of accumulation of inactivated because of accumulation of mutation, that type of gene is known as conventional gene. Pseudogenes, where the genes derived from reverse transcription of mRNA. So here mRNA, we know the central dogma, that is DNA is transcribed to mRNA and translated to protein, but here mRNA is reverse transcribed to form genes. Integration of that reverse transcribed DNA is known as processed genes. See here, homologous genes, genes which share a common evolutionary ancestors. What are homologous genes? So genes which share a common evolutionary ancestors. A pair of homologous genes has sequence similarities. So some of the sequence of the gene is same in a pair of homologous genes, but do not have identical nucleotide sequence. It is because the two genes undergo different random changes by mutations. So there may be cases of insertion of nucleotide, single nucleotide, so it do not have the identical nucleotide sequence, but they have some sequence similarities. So there are two categories of homologous genes as mentioned earlier, that is paralogous, where homologous genes present in the same organism that arose through duplication and divergence. So here you can see the paralogous genes and orthologous genes. In this, the homologous genes present in the same organism which arose through the duplication or divergence. Here gene duplication and divergence has resulted into gene G1, gene G2. So it has some similarities. But do not have identical nucleotide sequence. It is present in the same organisms. Example, myoglobin and globin genes of humans. And next is orthologous. It is a type of homologous genes in different organisms that arose through speciation and divergence. So here the same organism, here different organisms, and they have some different sequences, gene GA, gene GB. Here the gene GA and gene GB has the same evolutionary ancestor as said in homologous genes. Here also the genes are of having same common evolutionary ancestor, but after that, due to the speciation, they give separate species, and due to the speciation and divergence, the sequence has some different or has some dissimilarities. But here in paralogous, it is present in the same organism, and due to some duplication and divergence, the identical nucleotide sequences are not seen. Next is pseudogenes. Pseudogenes are false genes or unexpressed functionless, non-functional gene copies. They are derived from the functional genes itself. They are the type of evolutionary relic that indicates the changing nature of the genome. So genome has undergone different types of changes, which led to the genes to some of them become non-functional. There are abundance of pseudogenes and it is dependent on the rate of duplication and loss. Common features of multi-gene families: there are two classes: conventional, which is non-processed, and processed pseudogenes. As mentioned earlier, genes that has become inactive due to some accumulation of mutations like point mutation, frame shift mutations, that is known as conventional, which is non-processed or which is due to some accumulation of mutation by chance. Example, globin pseudogenes. Processed pseudogenes, the genes that derives from reverse transcription of mRNA and integration of that reversed transcribed DNA that is made through mRNA retrotransposition, is known as processed pseudogenes. So it has undergone some processing like reverse transcription, and that led to pseudogene formation. They lack introns and promoter sequences. So in our next class, we'll be discussing about how the new genes are acquired inside the organism, how the new genes are entering into the genome of the organisms. Thank you.